introduction, as mentioned, my name is Alex, and I'm a graduate student in the lab of Dr. Andreas Schultz. And today, I'll be presenting one of my projects, which is the optimization of a stable isotope assay for measuring AGAT activity. So creatine and phosphocreatine are key molecules that are involved in ATP regeneration and homeostasis. So when creatine is taken up by cells via the creatine transporter, it becomes phosphorylated into phosphocreatine through the activity of creatine kinases within the mitochondria and within the cytosol. This phosphocreatine can then be hydrolyzed to allow for the regeneration of ATP from ADP. So in this way, creatine and phosphocreatine act as buffers against imbalances in ATP levels in the cell and maintains its homeostasis. Now, in addition to obtaining creatine through supplements or through dietary means, creatine is also produced within the body in a two-step enzymatic reaction. In the first step, arginine and glycine are converted into guanidinoacetate acetate and ornithine through the activity of an enzyme called arginine-glycine aminotransferase, or AGAT. Guanidinoacetate acetate, in turn, is then converted into creatine through the activity of a second enzyme, guanidinoacetate acetate methyltransferase, or GAMT. Creatine itself is then non-enzymatically converted into creatinine, where it can then be excreted as a waste product. Now, in individuals with deficiencies in either AGAT, GAMT, or the creatine transporter, they develop a condition known as creatine deficiency syndrome, which is an inborn error of metabolism that occurs due to an inability to either produce or take up creatine. Patients with this condition have reduced levels of creatine within the brain, and they have symptoms including developmental delays, speech impairments, as well as seizures. Depending on the underlying cause of their condition, treatments for these individuals may vary. Now, there are several ways in which we can diagnose patients, prospective patients, with creatine deficiency syndrome. One way is through magnetic resonance spectroscopy, or MRS, which aims to measure the levels of metabolites within the brain and compare them to their expected baseline values. Doctors may also look at the levels of guanidinoacetate acetate, creatine, or creatinine within the blood or urine using tandem mass spec, or mass spectrometry, which identifies these compounds based on their mass-to-charge ratio. Finally, the most definitive method for diagnosing patients is through DNA sequencing of the gene in question to look for mutations within the sequence. However, under conditions in which these methods provide ambiguous results, alternative methods for confirming diagnosis may be required. And so one such way is through the use of a stable isotope-labeled substrate assay, which is used to measure enzyme activity. And so this assay was actually first developed back in 2003 by Verhoeven, where they measured AGAT activity in lymphocytes and lymphoblasts. And so for today's presentation, I'll be focusing on the optimization and the use of this assay for measuring AGAT activity in various mouse tissues as well as in human cell lines. But before I continue with the presentation, it's first important for me to establish what exactly stable isotopes are. So stable isotopes are different forms of the same element that differ in the number of neutrons present within the atom. So one example of this is carbon-12. So carbon-12 is the predominant isotope of carbon, and it contains 6 protons and 6 neutrons, therefore giving atomic mass of 12, and therefore the name carbon-12. Its stable isotope counterpart, on the other hand, carbon-13, instead contains 6 protons with 7 neutrons, therefore giving atomic mass of 13. Now, as I mentioned previously, tandem mass spec works to identify compounds based on their mass-to-charge ratio. And given that carbon-12 and carbon-13 have different masses, this allows us to differentiate between these two compounds. As such, we can then use stable isotopes as a way to track and measure the production of certain compounds within a cell or within a reaction. So here we're using a stable isotope-labeled substrate assay with stable isotopes in the arginine and glycine. So in the arginine, we have 2 stable isotopes of labeled arginine, as indicated here. So we have 2 stable isotopes of nitrogen-15. In the glycine here, we have 2 stable isotopes of carbon-13, followed by 1 isotope of nitrogen-15. Now, when AGAT uses these 2 substrates, it results in the production of normal or unlabeled ornithine, as well as the production of guanidinoacetate acetate that contains 5 stable isotopes. Hence, we give it the name penta-GAA. We can then track the amount of penta-GAA present in a reaction in order to quantify or measure the amount of AGAT activity in a system. So the first thing we want to do was to validate that the addition of these 2 labeled substrates can result in the production of penta-GAA. So what we did here is we performed the enzyme assay in mouse kidney homogenates, and we added in either normal arginine glycine or labeled arginine glycine, and measured the levels of both GAA and penta-GAA within the reaction. So in the top row, what we're seeing here is the amount of GAA present within the reaction. And so on the very leftmost figure, we have the baseline. So this is just the kidney homogenate in and of itself without any substrates added to it. And we can see that even without any substrates, we still do have levels of GA present, therefore showing that the kidney in and of itself does contain detectable levels of GAA. Now when we add labeled substrates, we have a significant increase in the amount of GAA being present in the reaction now. And when we add labeled arginine and glycine, we still do have GAA present, but we can see the intensity of the GAA present is similar to the baseline levels. Therefore, this shows that the addition of the labeled arginine and glycine does not contribute to the formation of normal GAA. In the bottom row, we're looking at the levels of penta-GAA in the systems, in the reactions now, and we can see that in both baseline and in the unlabeled conditions, we have no amount of penta-GAA being synthesized. Instead, we only have penta-GAA being synthesized when we add both labeled substrates within the reaction. So based upon this observation, this shows that when we add both labeled substrate, we're able to produce detectable amounts of penta-GAA. Taking the previous experiment one step further, we want to verify or validate that the production of penta-GAA was specific to having both labeled substrates. So to do this, we performed the assay in the combination with either both unlabeled substrates, both labeled substrates, or one of each, and we measured the amount of GAA or penta-GAA being synthesized. So in the first two lanes here, where we have one labeled and one unlabeled substrate, we do not have any levels of GAA or penta-GAA being synthesized. Instead, we only have penta-GAA being synthesized when we have both labeled substrates, and we only have normal GAA being synthesized when we have both unlabeled substrates. Furthermore, adding high levels of ornithine to either of those two reactions results in a significant reduction in the amount of GAA being synthesized. Finally, the panel on the right is looking at the amount of ornithine being synthesized in each of these different reaction combinations, and we see that they're very similar to each other, which makes sense because they all produce the same unlabeled ornithine. Now moving forward, now that we've validated that our assay in fact does work, our next goal was to optimize the reaction conditions for it by changing or by looking at the temperature, pH, and the assay duration. And so once again, this was done using mouse kidney homogenates, and our first goal was to look at the temperature condition. So what we have here is we're looking at the different temperatures on the x-axis, and on the y-axis we're looking at AGAT activity. So when we plot the data and fit it to a Gaussian curve, we can see the ideal temperature for performing the assay is at around 37 degrees Celsius. Moving on, we did the same thing with pH and looked at the pHs ranging from 6 to 10, and the ideal is around 7.6. Now with these observations, so an ideal temperature of 37 degrees, an ideal pH of 7.6, this makes sense because these are the conditions that are similar, that are found under human physiological conditions. And so therefore it makes sense for AGAT to act ideally under those circumstances. Finally, we also looked at the condition of time. So essentially, how long should we perform the assay for? And so we performed the assay for either 0, 30, 60, 90, or 120 minutes, and we're looking at the amount of penta-GAA being synthesized here. And so we can see here, this is a strong linear correlation between the duration of the assay and the amount of penta-GAA being synthesized. And therefore, this shows that the assay can be performed for at least 2 hours without any concern for substrate depletion. So now that we've both validated and optimized conditions, our first goal was to look at the kinetics of the AGAT enzyme in the mouse kidney. So to look at enzyme kinetics, we keep the concentration of one substrate constant while we modify or we change the concentration of the other. So on the panel on the left here, we're looking at the kinetics of AGAT for glycine. And so to do this, we keep the concentration of arginine constant while we vary the concentration of glycine over a range of 0 to 15 millimolar. We can then fit the data to a Michaelis-Menten equation and calculate the Km and Vmax of it. So the Km of AGAT for glycine is around 2 millimolar with a Vmax of 6.48. And we do the same thing for arginine. We can also calculate its kinetics as well, giving us a Km of 2.6, and a Vmax of 2. So while there isn't any data in the literature regarding kinetics of AGAT in mouse tissues, the data we've obtained here is quite similar to that obtained in AGAT for humans and rats. So this gives us some degree of confidence that our results are in fact accurate. So moving forward, now that we've done all of this optimization testing, we next want to look at AGAT activity in mouse tissues. So specifically, we looked at AGAT activity in the mouse kidney, heart, brain, liver, and muscle. And so we can see in the data here is that we have the highest amount of AGAT activity in the kidney, followed by the brain and liver, with the heart and muscle having AGAT activity that is below our limit of detection. So what's actually interesting to note here is that even though the liver does contain AGAT protein, the amount of AGAT activity that we actually detect is very minuscule, basically almost nothing. And so we want to look at this and basically explore why this was the case. So what's interesting to note is that the liver actually contains high levels of an enzyme called arginase. And in fact, arginase is only expressed in very few tissues, with it being expressed at high levels in the bone marrow and liver, medium levels in the spleen and testes, and low levels in the skin. But what exactly is arginase? So arginase is an enzyme that cleaves arginine in order to generate ornithine and urea. And so as a consequence of this, having high levels of arginase can affect AGAT activity in two main ways. The first, the high levels of arginine results in depletion of arginine. And given that arginine is a substrate needed by AGAT, this results in a reduction in its activity. The second way in which arginase affects AGAT activity has to do with one of the products it produces, ornithine. Now, as I mentioned previously, arginine and glycine are used by AGAT to synthesize GAA and ornithine. And ornithine is a strong competitive inhibitor of AGAT. As such, when we have high levels of ornithine present in a reaction, this results in a reduction in AGAT activity. So in summation, arginase essentially inhibits AGAT activity in two main ways. The first is through depletion of arginine, which is a substrate needed by AGAT, and the second mechanism is that it causes an increase of ornithine, which is an inhibitor of it. Now, to deal with the high concentrations of arginase present in the liver, we counteracted its effects with a compound known as nornoha, which is an arginase inhibitor. Now, to test how effective arginase essentially is to test how effective nornoha is at inhibiting arginase, we performed this reaction in mouse liver homogenates, added different amounts of nornoha, and we looked at how much ornithine and arginine are present in the reaction. And if you look at the panel on the left, we can see that with increasing amounts of nornoha added, we have reducing amounts of ornithine being generated with a kind of plateauing at around 500 micromolar. In contrast, if you look at the graph on the right, you see the opposite phenomenon in which as we increase the amount of nornoha present, we have increasing amounts of arginine now being rescued within the reaction. As such, this shows that nornoha is an effective inhibitor of arginase as it effectively reduces the amount of ornithine present as well as increasing the amount of arginine in there. Finally, before that, when we redo the enzyme reaction now in the mouse liver samples, we now have a more accurate activity of 0.48, which is approximately a 160-fold increase in AGAT activity compared to the liver without nornoha addition. Finally, we also looked at AGAT activity in various patient-derived cells. So what we did here is we measured AGAT activity in lymphocytes, lymphoblasts, and fibroblasts. Now, the reason why we chose these cells is that lymphocytes and lymphoblasts are derived from blood, and fibroblasts are derived from the skin. And these two sources are most easily accessible, and therefore, this makes it easier to do this assay for potential diagnostic purposes. And so what we can see here is that we were able to measure AGAT activity in both the lymphocytes and the lymphoblasts, with the lymphoblasts having significantly higher levels of activity. And unfortunately, we were not able to detect any AGAT activity at all in the fibroblasts. So with that, in today's presentation, I've demonstrated the use of a stable isotope assay for measuring AGAT activity in various mouse tissues and in cell lines. With this, we were able to measure the enzyme kinetics of AGAT in mouse kidneys, and we also observed the phenomenon in which high levels of arginase in certain tissues may impact our recordings or measurements, essentially. Moving forward with this project, the next goal is to measure AGAT in different mouse genotypes, specifically mice are either heterozygous or mutant for AGAT, to see how having a heterozygous mice might affect the ability of AGAT to perform its reactions. And then finally, we're also looking to measure AGAT activity in more human-relevant cell lines, so control lymphoblasts, as well as control in human patient tissues, with the goal of establishing a comprehensive baseline, and this can aid also in diagnostic purposes. And so with that, I'd like to thank the members of my lab for their support and assistance. Unfortunately, none of them are here, so it's just me. And then finally, I'd like to thank the ACD and everyone here for listening.
Thank you so much for that presentation, and I don't see any hands up yet, so I do have a couple questions. So I think earlier this morning, one of the things that was brought up for like the newborn screening panel and stuff is problems across different instruments, right? And so if this is mass spec and with isotope, I'm assuming this could be applied across different type of mass spec instruments then in theory, like for this test to see?
Theoretically, yes, you should be able to do it as long as you calibrate the instrument properly and you know kind of how to detect the compounds, then yes, you should be able to do it.
Okay, and is this only something that you have looked at from like the homogenized tissue specific, or is this something that you could see being moved over to like the dry blood side, like being able to test from that aspect, or even like urine, or I guess not, since it's not in high abundance in that tissue?
To some extent, the ability of this assay depends on the amount of enzyme present, and so we would need a sample that would actually contain the enzyme, and for blood samples that might be a bit difficult, especially because they're dried, and therefore that might cause issues with the enzyme activity, essentially. So there's kind of that degree of limitation.
Perfect, thank you. And are there questions for me? I do, okay.
Cool, Alex. Great talk. So the homogenates, so with all the organs, are you preparing homogenates and then measuring it in total homogeneous solution?
Yeah, so for mouse tissues, we typically just use whole homogenates, and then we normalize the data based on the amount of tissue we've used, just simply because it's difficult to get accurate protein quantifications from tissue homogenates.
Cool. And do you know, is arginase expressed in hepatocytes or in endothelium?
Are you referring to it as a cell line or just in general?
Or so when you show that the arginase activity is really high inside of the liver, do you know what cell type it's arising from?
Not particularly. When we do our assays, we don't really have the ability to just look at cell type specifically, but just as a whole, it just happens to have high levels of arginase.
Yeah, it's interesting because the AGAT expression is from the endothelium of the liver, and the hepatocyte doesn't actually have much AGAT, and I wonder if that's probably the reason why.
It could be, but yeah.
It's very cool.
All right, and okay. Did you have some more questions?
Alex, wonderful job. When you say that you need controls, can that be your optimized wild-type lymphocytes diluted in PBS, dried on dry blood spots, and that becomes a standard?
Sorry, do you mind repeating again?
I said that you can take your standard lymphocyte cell line and you can dilute it, and that can be a standard for your AGAT, anywhere you want to do the assay.
Theoretically, yes. We can also use, ideally we would like to use control lymphocytes as well as a control, so yeah.
All right, do we have any other questions? I do have just one more then. You had measured for the penta, GAA. Did you ever notice in there by chance like a quad GAA, that it was occurring from kind of that nitrogen, like where they connect, that it was getting substituted out from the nitrogen in the other amino acid that's unlabeled?
So for the reaction that we're doing here specifically, we've only seen penta-GAA, and just the way the reaction works, we never uncovered or we never identified any situation in which we only had 4 labels.
So it's consistent with the penta?
Yes.
Every time? That's great. Okay, Heidi's got a question.
Since we have a little bit of time, I was following along, and I kind of lost you on the slide where you were showing glycine versus arginine kinetics and what the significance was that I was supposed to glean from that.
To some extent, there's not much significance with regards to, for this data, there's not much significance with regards to the families, unfortunately. This is just purely for scientific knowledge.
I'm following you. So what is it? What's the scientific knowledge? I'm not understanding this.
So this is just to understand. So we know what the kinetics of AGAT are in different other species, so human and rats, and this is just to basically make sure or validate that the same AGAT had the same kinetics in mice as well. And this is also just more of just looking for knowledge. There's not much else beyond that.
Okay, thank you.
Sorry, this is a bit of a complicated slide for this audience.